Hi everyone, welcome to my channel. In this video we will talk about amino acid catabolism. So we are going to get an overview that how the proteins are broken down into amino acids and how these amino acids are broken down to nitrogen and how it is excreted. So we know in our stomach and in our intestine, with the help of several enzymes such as chymotrypsin, trypsin, pepsin, carboxypeptidase, which degrade proteins into the simplest form, which is amino acid. The and this amino acid is absorbed into intestinal epithelium cells and it moves into the blood, and then it is actually used to make muscle protein or other aspects, but in this video our focus is how protein breakdown occurs at the molecular level. So proteins are broken down into amino acids, that is the basic constituent of the protein. Now this amino acid is further broken down. The amine part of the amino acid generates ammonia and further excreted as urea, whereas the carbon skeleton of the amino acid is recycled used in several biosynthetic pathways such as TCA cycle. Now in this video we will look at the overview of amino acid catabolism. So the hub of amino acid catabolism is the liver. Imagine there are four amino acid 1, amino acid 2, amino acid 3 and amino acid 4. Each of the amino acid would donate their amino group to a substance which is ultimately glutamate. So it would generate glutamate. From glutamate the ammonia would be generated and the alpha ketoglutarate would be the side product of it. Now this ammonia is extremely harmful and toxic, so the ammonia will be converted into a less harmful form which is actually urea, and urea is the form which is excreted. So the process of conversion of several amino acid to glutamate is known as transamination, where this amino acid donate their amine group to glutamate. Now from glutamate the process of generation of ammonia is known as deamination, and lastly the urea cycle generates urea from the ammonia. So this is the overall workflow of amino acid catabolism. So let's look into this process in more details. So the first step is nothing but the breakdown of amino acid into glutamate or funneling it into glutamate, and it happens inside the liver hepatocyte. So in the liver hepatocyte any amino acid is converted into a keto acid in a process known as transamination, which is a amine group transfer reaction. The key prosthetic group of the enzyme aminotransferase is the pyridoxal phosphate, and the aminotransferase enzyme plays an important role in transamination reaction, and the pyridoxal phosphate of this enzyme plays a critical role in the process of transamination. So what happens within liver, the glutamate, with the help of glutamate dehydrogenase, the amine group NH3 group will be transferred with the help of a enzyme glutamate dehydrogenase, and it will be form an intermediate which is alpha immunoglutarate, and ultimately it will produce alpha ketoglutarate. So from glutamate the amine group will be transferred with the help of glutamate dehydrogenase, and it will form the alpha ketoglutarate, and the intermediate product of this reaction is alpha immunoglutarate. In this process one molecule of NADP will be converted into one molecule of NADPH or reduced NADP. In this process the ammonia, which is toxic, will be converted into urea, which is a less toxic or less harmful, and it will be excreted out. So what happens in the urea cycle? Urea cycle occurs both in mitochondria and in cytosol. In the first reaction of the urea cycle, the ammonia, with the help of the enzyme carbamyl phosphate synthetase, as its name implies, it requires the ATP molecule as an energy source. and the ammonia will first form the carbamoyl phosphate. The carbamoyl phosphate now reacts with ornithine and it will form the citrulline. Then citrulline will attach with aspartate molecule and it will form argininosuccinate. Argininosuccinate will produce arginine and arginine will be converted into ornithine. So the ornithine will be again recycled, and in the reaction where arginine is converted into ornithine, urea will be formed. So this is the steps of urea cycle. So what we have found that amino acids lacking storage forms are continually degraded as proteins turnover. The alpha amino group is removed and the carbon skeleton of the 20 amino acids are funneled into 7 key molecules like pyruvate, acetyl-CoA, acetoacetyl-CoA, alpha-ketoglutarate, succinyl-CoA, fumarate and oxaloacetate. So what is the glucogenic amino acid which is degraded into pyruvate, alpha ketoglutarate, succinyl-CoA, fumarate or oxaloacetate, allowing glucose synthesis via gluconeogenesis. Ketogenic amino acids are degraded into acetyl-CoA or acetoacetyl-CoA, forming ketone bodies or lipids. Classification: solely ketogenic amino acids are leucine and lysine. Both ketogenic and glucogenic হচ্ছে isoleucine, phenylalanine, tryptophan and tyrosine. Solely glucogenic হচ্ছে the remaining 14 amino acids. So this is the summary of the amino acid catabolism process. Thank you. All the best, my dear students.